Hello aspirants, welcome to Vital Science Institute. In today's video, we will discuss about Trp Operon, a major regulation of gene in case of prokaryotic system. Now, before going into the depth of it, let's understand a few points over here. E. coli, Trp Operon, it consists of 5 structural genes, namely Trp EDCBA. Coming to this picture, let's see. We have a set of genes where we can find there are structural genes like E, D, C, B and A. Upstream of these sequences, these structural genes, lies our leader sequence. Apart from leader sequence, we have a operator site. And there are another set of genes which is called trpR, which is associated with repression. Now, it can be located either upstream or downstream of this entire segment of gene. Now, the fact is, when it comes to, let's come to the second point. The percentage of Trp within a given protein is about 1%, which is very less. And the most frequent amino acid within any kind of protein is leucine. Just for information. For the synthesis of tryptophan operon, the major enzyme which is required is tryptophan synthase. This is the one and only enzyme which is required for tryptophan biosynthetic pathway. As you can see over here, tryptophan synthase is the major enzyme which is required. And another important point we have to understand from this part is the precursor of tryptophan should be chorismate. The precursor of tryptophan should be chorismate. The fact is, what type of question can come from this segment? Now let's see. As we all know, tryptophan concentration, when the percentage of tryptophan within a cell is less, what will happen? Then we need to synthesize more tryptophan via Trp Operon, and for that we need to enhance the tryptophan synthase enzyme. As you can see from this graph, when the tryptophan synthase activity is high, the Trp, the overall Trp level should be low. That means tryptophan synthase activity high. When it will occur? When the overall tryptophan level is low. Similarly, when the overall tryptophan level is high within a cell, then the amount of tryptophan synthase will be low. So, coming to this point again, try to remember, tryptophan synthase is one of the most important enzyme in this entire pathway. So, if tryptophan synthase activity is high, that means the overall tryptophan level within the cell should be low, and when the tryptophan synthase activity within the cell is low, that means the tryptophan level within the cell should be high. So that means the tryptophan synthase activity and the concentration of tryptophan, these two factors, they are inversely proportional. Question can come from this part B. The same thing is written over here. The amount of tryptophan synthase within a cell is inversely proportional to the concentration of Trp which is present within the cell. Now, coming to our second set of points. Before Trp Operon, we have came across lac operon. Now, lac operon, unlike T operon, it generates energy. That means it generates what? Glucose. So, a certain amount of energy is being generated within the cell. But when it comes to Trp Operon, the overall mechanism of Trp synthesis, it requires a huge amount of energy. That is why the overall mechanism is an anabolic process. Note it down, is an anabolic process. Coming to our next point, which is very important, the overall Trp Operon. The overall Trp Operon is negatively regulated by two distinct mechanisms. The Trp operon is negatively regulated by two distinct mechanisms. One, repression, another attenuation. Unlike lac operon, we have only one kind of repression by a mechanism called repression. But here we will require two distinct mechanisms. One is a repression, another one is an attenuator. Now why this mechanism? This information is taken from Weaver. Repression, when only trpR, that means Trp repression is present, it can repress the overall Trp operon 70-fold. It can repress the overall Trp operon 70-fold. And if attenuator is separately applied, then it can repress 10-fold. Now that means if repression by means of trpR and attenuation, both these factors are mixed, are combined or conglomerated, then what is happening? Then the overall repression is reaching up to 700-fold. That means 70 into 10, 700-fold increase in case of repression is observed when both these factors are involved. This is a very important information. It can come in part C. Next, as you can see, we have provided the same kind of information over here. Unlike lac operon, repression by Trp operon is extremely big. That is why it requires the presence of both attenuation as well as repression. Very simple. Next, attenuation is primarily based upon riboswitch within the leader sequence. Now before that, I want to describe a few things. How repression by trpR takes place. Let's see. Here lies our trpR gene. The mRNA is being synthesized by means of transcription, leading to the formation of a aporepressor monomer. trpR in its monomeric state is a aporepressor. This aporepressor monomer, then it will form a dimer. That means trpR is forming a dimer. It cannot work singly. For its activity, it will require tryptophan. Here tryptophan is acting as a co-repressor. Therefore tryptophan, when it will bind with the aporepressor, it will form a complex, the repressor dimer. This complex shows a high affinity. This complex, it shows a high affinity towards our promoter site. This promoter operator site, they are kind of overlapping. So that's why I am stating it as a promoter site, and it is located upstream of our leader sequence. It is located upstream of our leader sequence within the operon. So this is how the overall mechanism of repression takes place by means of trpR. Now coming to this important point. Repression by means of trpR is complete. Next, we will discuss about attenuation. Attenuation is primarily based upon riboswitch within the leader sequence that functions co-translationally in the Trp operon. Now, what is riboswitch? Riboswitch is a kind of mRNA that is associated with regulation, where it can change its shape based upon the binding of certain proteins. Here, the riboswitch occurs within the leader sequence. Here, the riboswitch occurs within the leader sequence of our operon. Let's analyze our leader sequence. Within the leader sequence, we have 4 regions. 1, 2, 3, 4. 4 regions are taken. Now, these 4 regions they are kind of intercomplementary. They are kind of intracomplementary. That means 1 can pair with 2, 2 can pair with 3, and 3 can even pair with 4. But the pairing between 1 and 2, it shows maximum amount of hydrogen bond, and the pairing between 3 and 4, it shows maximum amount of hydrogen bonds. So these two pairings are very strong, as you can see in the figure. This is kind of a attenuation, and the attenuator loop within the Trp Operon leader sequence is mainly formed between region 3 and 4. Again note it down, the attenuator loop formed within Trp Operon leader sequence takes place between region 3 and 4. Here it is mentioned why the complementarity is located, and the overall leader sequence is near about 140 nucleotides in length. The overall leader sequence is near about 140 nucleotides in length. So attenuation problem is being solved. Fact is, how come this overall mechanism is being regulated? We will discuss this regulation in 2 steps, at transcriptional level and at translational level. That means involvement of ribosome and involvement of RNA polymer. We know this is a bacterial system, therefore transcription and translation, both of them are occurring simultaneously. That means it is a co-translational mechanism. So we have to understand how the overall mechanism of regulation is being conducted when it comes to attenuation at high levels of TRP and low levels of TRP. First, we will start with ribosomal activity. That means during translation, what is happening? Now, attenuator riboswitch, that means the leader sequence, it bears 2 distinct checkpoints at region 1. What are the checkpoints? Let's see. Just like cell cycle. One is UGG codon at position 10 and 11 of the region 1. Now leader sequence. Leader sequence, it has a start codon which is AUG, and within the leader sequence lies our UGG codon at position 10 and 11. That means that codes for tryptophan, and the second should be UGS stop codon in between regions 1 and 2. These 2 factors are to be taken under consideration. Now let's see. When the tryptophan concentration is low, suppose the tryptophan concentration is low, what is happening? Let's come to this picture. See, here the tryptophan concentration is low. We have our leader sequence R1, R2, R3, R4, followed by our structural genes. Now transcription, as we are discussing over here, only we will focus on translational mechanism. Here transcription has arrived, has taken place, followed by translation. So the entire, first of all, one thing we must remember, RNA polymer, it will synthesize the entire leader sequence. RNA polymer will synthesize the entire leader sequence in both the cases, irrespective of high or low level of tryptophan. Again, note it down. RNA polymer will synthesize the entire leader sequence irrespective of high or low level of tryptophan. Then come to the point. Fact is, when transcription has taken place, we have got our mRNA. Now the condition is tryptophan level within the cell is low. Ribosome has bound to it. Ribosome will bind at AUG start codon of the leader sequence region 1. So it will start synthesizing. So we will get a peptide stretch. But there lies a problem. as we have seen that within the leader sequence, we have UGG codon. Within the leader sequence, region 1, we have UGG codon at 10 and 11 position. This is the criteria. So, if tryptophan level within the medium is low, then what will happen? At codon number 10, the ribosome won't be able to incorporate tryptophan because tryptophan level within the medium is low. So, the ribosome will stop. It will stall at region 10 within the mRNA of leader sequence. Therefore, we will get a peptide. We will receive a peptide which should be approximately 9 amino acids in length, which should be near about 9 amino acids in length. Because at the 10th amino acid there will be UGG, and as a result we will not get any kind of tryptophan from the medium. Coming to another phenomenon, which is high tryptophan level. When the tryptophan level is high, what will happen? As you will see, transcription and translation, they are occurring simultaneously and all the leader sequence have been synthesized at mRNA level. Now, ribosome has bound to the AUG start codon. It will start synthesizing the peptide as usual, and as tryptophan level is high, therefore, region 10 and 11, which encodes tryptophan, will easily be passed because we have huge amount of tryptophan already present within the cell. They will receive the tryptophan and the peptide stretch will occur. But there is a limitation. At the end of region 1 and at the beginning of region 2, at the end of region 1 and at the beginning of region 2, at this region, there lies a stop codon, which is UGA. Now, if there lies a stop codon, then what will happen? The ribosome, after synthesizing, when it will receive the stop codon, it will get released. The ribosome will get released. So, we will receive a stretch of leader peptide, which is near about 14 amino acids in length. That will start with our methionine and end with our serine. This is the regulation at translational level. This is the regulation at translational level. Let's repeat it. So, when TRP concentration is low, what is happening? Leader sequence, it bears our AUG start codon, as I have stated. Synthesis will start. As usual, the mechanism of synthesis will start. It will stop just before the 10th codon, UGG. Cause, what is the reason? Just stated. Cause it lacks TRP. The medium lacks TRP. So it will stop at the 10th region. Again, ribosome gets stalled at region 1 of the leader sequence at 10th codon due to lack of tryptophan. And then the peptide will be near about 9 amino acids in length. Because at the 10th codon, we are not getting any kind of incorporation of amino acid. So, this will be the regulation mediated at translational level. Next, the phenomenon when TRP concentration is high, what will happen? The leader sequence will start synthesizing the peptide as usual, just like the previous one, as we have AUG codon. Now, it will easily pass the 10th and 11th codons because we have sufficient amount of tryptophan within the medium. So, they will eventually be incorporated. The peptide synthesis will stop at stop codon UGA. At stop codon UGA, the synthesis the peptide synthesis will stop at stop codon UGA. Note it down, between regions 1 and 2. Now as usual we all know that ribosomes will dissociate. At stop codons the ribosome usually dissociates. This is the basic phenomenon. So at translational level the overall regulation is being mentioned when the tryptophan level is low and when the tryptophan level is high. Now, the most important point, what is happening during transcriptional level? This is a very important phenomenon. Let's see. Suppose TRP concentration is high. TRP concentration within the medium is high. As usual, our ribosomes will come. We have our mRNA, and as I have mentioned just before, that all the sequences within the leader will be transcribed up to region 4. All the regions within the leader sequence will be transcribed up to region 4. Hence, we have received the entire leader sequence in mRNA form. All the regions are there. Now, region 1, we have our AUG codon. Therefore, ribosome will bind to it and it will start synthesizing. Now, when ribosome is synthesizing region 1, and just previously we have mentioned that when tryptophan concentration is high, we are getting ample amount of tryptophan to bind at region 10 and region 11. So therefore, they will eventually be passed and finally the ribosome will be released at stop codon UGA. By this time, what will happen? By this time, what is happening? Synthesis is occurring as usual. Ribosome is getting released. Now we have the mRNA. That means, suppose this is our mRNA. Here the ribosome is undergoing synthesis. Here it is encountering our UGA stop codon. Now rest of the regions are there. Region 2, region 3, region 4. These regions are there. And this is the mRNA segment. And what we have previously known, that region 3 and region 4, in fact all the regions, they are intra-complementary. That means when translation is occurring at region 1, we should be observing a kind of stem loop formation. We will be observing a kind of stem loop formation in between region 3 and region 4. That means this overall structure will be same, but the sequence will change at 3 and 4. Here we will observe a stem loop structure. That means first pairing occurs between region 3:4, and this is called our attenuator loop. This is called our attenuator loop. Now, many aspirants can ask that this overall mechanism is happening, it's well and good, but RNA call has already passed this region. RNA call has already transcribed this region, and so they are supposed to enter our structural genes. they are supposed to enter the structural genes by that time. Then why it is not happening? That means there must be certain mechanism that is kind of removing the RNA pol from the segment of DNA before structural genes are to be synthesized. That means, I am repeating it, RNA pol, just look at this, RNA pol has already synthesized 4 regions of the leader. So, based upon the fact, it can easily enter the structural gene segment and it can synthesize the mRNA for structural genes. But why not it is happening? Because there must be some mechanism that is somehow removing the RNA pol from the DNA. And what mechanism is that? Apart from attenuator loop, when at UGA, the ribosomes are released, then what is happening? We are getting free region 1 and region 2. We are getting free region 1 and region 2. Therefore, first pairing between 3:4, which is the attenuator loop. Second pairing, after the removal of ribosomes, we are getting 1:2 pairing. This 3:4 pairing plus 1:2 pairing, both is initiating the release of RNA pol. Very important point. These two are initiating the release of RNA pol from the DNA. Hence, none of the structural genes can be synthesized. This is a row independent termination of RNA pol, and this is happening when the TRP level, the tryptophan level, is high. Let's go through these segments. mRNA, when tryptophan concentration is high, what is happening? mRNA is synthesized up to region 4. As simple, mRNA is synthesized at region 4 by RNA pol. First pairing of mRNA between regions 3 and region 4 is happening. Cause leader sequence already synthesized. 3 and 4, they are intra-complementary, and hence 3 and 4 will pair with each other. Next, ribosome is released at stop codon. What is the stop codon? UGA. Ribosome is released at stop codon, and where it is located? Between region 1 and 2. Now, when the ribosome is released, what is happening? Second pairing. This is the first pairing. This is the second pairing. Second pairing is occurring between newly freed. Why it is termed as newly freed? Because region 1 of the leader sequence is being occupied by the ribosome. When ribosomes are released, they are freed. Hence, newly freed regions 1 and 2. Now, 3:4 pairing plus 1:2 pairing. One is attenuator loop. These two pairing will help in the release of RNA pol. These two pairing will help in the release of RNA pol. And as a result, no synthesis of mRNA in the region of structural genes will take place. as RNA ball is being removed in a row independent manner. This is the mechanism at transcriptional level how the regulation takes place when tryptophan concentration is high. Let's see. Tryptophan abundance. As you can see, pairing has occurred between 1 and 2 and 3 and 4 based upon its complementarity, and hence the ribosome has fallen off, and this kind of pairing has helped the ribosome, this kind of pairing has helped the RNA ball to be released from the overall DNA. Hence, no structural genes are to be synthesized. Now coming to our second point, tryptophan concentration low. Now let's see. Tryptophan concentration, TRP concentration is low. That means when tryptophan concentration is low, we are getting our overall region 1, 2, 3, 4, E, D, C, B, A. This is overall structure. Now, transcription has occurred. Due to transcription, what is happening? We are getting our mRNA. And as usual, ribosome has bound to it, that will initiate the process of protein synthesis. Now, tryptophan concentration low. That means this peptide, this overall peptide will be 9 amino acids in length, because we all know why 9 amino acid, as we have discussed previously. Why 9 amino acids? Because ribosome has stalled at region 1 of the leader sequence because of low concentration of tryptophan. That is why it should be 9 amino acids in length, and we have low tryptophan. So no tryptophan will able to bind with the 10th and 11th position of this mRNA. So ribosome will be stalled. Now we are getting the mRNA. We are getting the mRNA up to suppose leader sequence. That means all the regions within the leader sequence have been synthesized at mRNA level. Ribosome has stopped at position 10. Ribosome has stopped at position 10. Rest of the mRNA is being synthesized. Now you see, the ribosome, as it has stopped, take a deeper look into it. This is supposed to be the region 1. When region 1 of the mRNA within the leader sequence is blocked, sequence 2 is free, region 2 is free, region 3 is free. As a result, when 1 is blocked, region 2 is free. Therefore, instead of pairing between 3 and 4, as 2 and 3 are also intercomplementary with each other, then what will happen? This ribosome is there and it has been stalled, and we must observe another stem loop. We must observe another stem loop formation between region 2 and 3. As a result, region 4 is free within the mRNA. Therefore, in this case, when tryptophan level is low, as ribosome has stalled at region 1 of the mRNA within the leader sequence, region 2 and 3 are free, and region 2 and 3 they will pair with each other. When 2 and 3 will pair with each other, then 4 is free. As a result, neither 1:2 or neither 3:4 pairing is occurring. That means RNA pol will not be released. RNA pol will not be released. Very important point. Cause the pairing is occurring between region through this. As a result, what will happen when pairing between 2 and 3 is occurring? RNA pol will eventually synthesize all the structural genes, and hence tryptophan will be synthesized. This is how the regulation takes place when the tryptophan level is low. See, tryptophan concentration low, that means mRNA is synthesized up to region 4 by RNA pol. Typical example, ribosome blocking at region 1. Why? Because tryptophan level is low. Tryptophan level low means at 10th and 11th we are not getting tryptophan. When tryptophan is not there, ribosome is stalled. As simple as that. So ribosome blocking region 1 at position 10 and 11 due to lack of tryptophan. Region 2 of the leader sequence within the mRNA is free. When region 2 is free, hence pairing between region 2 and 3 will take place. And region 2 and 3 pairing is not kind of any attenuated loop. Therefore, it will not initiate any kind of termination of RNA pol. As a result, all the structural genes EDCBA will be synthesized by the RNA pol. Now, another question that can occur, suppose we have our mRNA. Some of the aspirants might think that the ribosome is already blocking region 1. Then how come this EDCBA structural genes are to be synthesized? Very important question. Now, we have to understand that all of these structural genes, their mRNA basically, it bears RBS sequences, that means ribosome binding site. Therefore, separate ribosomes will bind to all of these structural genes, and hence the proteins will be synthesized. And as the enzymes are being synthesized from these structural genes, tryptophan synthesis will occur. This is how the overall mechanism regarding tryptophan operon at transcription and translational level is regulated. Tryptophan starvation, that means tryptophan level is low. Here ribosome stalls within region 1 at position 10, and we are getting a pairing cause 2 and 3 they are kind of intra-complementary. 2 and 3 are pairing with each other, and region 4 is free, and hence region 4 is free, and it is 2 and 3 pairing is not associated with any kind of termination, hence RNA pol will perform their activity. This is how the mechanism will take place regarding tryptophan operon. I hope this video will find you with proper conception, and if you find this video interesting as well as informative, do share with all of your batchmates. And always remember, stay focused, build concepts and track progress. B. Shanku Banerjee, founder and educator at Vital Science Institute, signing off.